Okay, welcome back. Let us keep going together to explore some of the multi-layered information capacities of DNA. We've already explored some. We'll explore just a couple more areas to hopefully bring the point home. And as I said, to me, this is really the most fascinating set of lectures in the entire course because it is just unfathomable, the complexity that resides within the DNA code. So another multi-layer of information, another code within a code, is the regulation of alternative splicing. And we've already covered together what alternative splicing is, but let us just review. We said that when the DNA is transcribed into RNA, we have exons and introns. The exons are the regions that will be expressed as protein. And so we see that when the RNA is made, you have exon 1, exon 2, exon 3, exon 4, exon 5. Well, there can be selective splicing to make different messenger RNAs that will then make proteins. So for example, the left-hand mRNA takes all of the exons, exon 1, exon 2, exon 3, exon 4, exon 5 that are color-coded, and makes an mRNA that then gets translated into protein A. But we can alternatively splice that same RNA by, for example, taking out exon 3. That will no longer be expressed. Remember, exon stands for expressed region. So now I have an mRNA that contains exon 1, exon 2, exon 4, and exon 5. And when that is translated into a protein, that is a different protein, protein B. And same thing with protein C, except that now it's exon 4 that is missing. And we've already seen that alternative splicing is actually much more complex than that, because what we can do along the way is, for example, duplicate exon 3 and maybe duplicate exon 4 and drop exon 5. So there is a dizzying array of combinations that could be made from the same primordial RNA molecule. And so it is an ultra efficient system that with the same piece of DNA, through the mechanism of alternative splicing, we can make multiple different proteins. In this case, you see the simplest illustration, schematic illustration of alternative splicing, giving us protein A, protein B, and protein C. Now, is this process happening haphazardly? No, of course not. It is not happening haphazardly. It is a very tightly regulated process. And where does the information for this come from? Once again, it comes from the DNA. So the DNA at the same time carries the information that codes for the amino acid sequence to make protein A, protein B, protein C, etc. And it carries the information for how and when to splice the RNA to make the proteins that are needed. So this is a very important second layer of information, which is the layer that controls proper RNA splicing. Now, I just want to call your attention to something that we will need later. All of these spliced proteins, protein A, protein B, protein C in this case, they all are from one DNA region. They begin and end in the same place. Begin, say, with exon 1 and end with exon 5. Just please keep that in mind, and we'll see why that's important in a minute. Okay, so now let's look at another code within a code. We're going to call this regulation of overlapping coding regions. Now, as if the vast array of splicing possibilities was not complex enough, it turns out that DNA does something equally amazing or even more amazing, which is that there are overlapping coding regions And what that means is, let's say I need to code for protein A and protein B, and there's a string of bases that codes for protein A. Then I could start maybe in the middle of that string, take the second half and add on to it some more bases and make a new string that codes for protein B. So part of the bases that code for protein A also code for protein B. And now this is different than alternative splicing because remember we said with alternative splicing, everything has the same reading frame. What that means is the RNA begins in the same place, ends in the same place, but is just internally spliced in different ways. Overlapping coding regions means that there are different reading frames. Somehow, when we are going to transcribe that DNA into RNA, there's an embedded layer of signals that says begin here and end here to give me this protein. So let's look at the bottom of the slide. There's information that says the protein I want, and in this case the protein will be a sentence made of 3-letter words like the 3-letter codons. The cat ate the rat who was sad. Alternatively, I could want a different protein sentence, which is the rat who was sad did not see the cat. And you see that there is an overlapping region there, and that overlapping region is in red, the rat who was sad. So somehow DNA is structured such that the bases in the DNA would be the cat ate the rat who was sad did not see the cat. And I can begin and end to make the first sentence, or begin and end to make the second sentence. And that information for the overlapping reading frames, where do I begin and where do I end, so that I could use part of the nucleotide sequence for one protein to make a second protein, that is a very, very complex set of regulations. That requires a tremendously fine balance. And that is precisely what this paper here mentions, that this was the beginning of the discovery of this sort of dual coding genes. And let us begin reading part of the abstract together. So it says coding of multiple proteins by overlapping reading frames. And again, this is not the same reading frame but spliced differently, but overlapping reading frames like we just covered. Coding of multiple proteins by overlapping reading frames is not a feature one would associate with eukaryotic genes. Eukaryotic genes are our genes, genes in a nucleus. Indeed, codependency between codons of overlapping protein coding regions imposes a unique set of evolutionary constraints, making it a costly arrangement. Yet in cases of tightly co-expressed interacting proteins, dual coding may be advantageous. Here we show that although dual coding is nearly impossible by chance, please focus on that. Here we show that although dual coding is nearly impossible by chance, a number of human transcripts contain overlapping coding regions. Let us stop here. Over time, this has only grown in importance. And so you see again this multi-layered function, this code within a code, and these authors are stating outright that to get this sort of intricate structure, that is essentially impossible by chance. So I think you're now beginning to get the idea that the DNA is so intricately structured that it literally defies imagination, and that it is not just one code embedded within the bigger code, but that it is really multiple layers of information. And so here from a Wikipedia article on Edward Trifonov, as I had mentioned earlier, he was really one of the pioneers of discovering this multi-layering of information. So he just, this article goes through and talks about some of these multiple layers, some of which we've covered. So for example, the layer that is in the DNA sequences, that is the chromatin code, and that is the set of rules responsible for positioning of the nucleosomes, and that is those spools that the DNA is wound around. Then the codes that set up particular RNA sequences. So first, of course, there's the triplet code that codes for the amino acids. We know that. Then we've talked about the splicing code, which is responsible for the different genes that are produced by differential splicing. Then the framing code that we just finished talking about, that basically where do we begin and where do we end, where is the reading frame, and a subpart of that is the notion of alternative reading frames that are sometimes overlapping. The translation pausing code, things that slow down or pause the translation of the RNA, and we've already talked about that, that allow time for the protein to fold properly, and we've seen that a lot of that is governed by the use of the synonymous codons. And so again, that goes into the protein folding codes that allow portions of the protein to be formed at different speeds so they can fold properly, and so on and so forth. And again, we can't cover everything, but we are just using this because these are all papers that were contributed to by Trifonov. So you see how much he personally has contributed to uncovering many of these codes, and it's a nice way for us to review some of the things that we've already talked about. And so, of course, each time one of these discoveries is made, interestingly, it filters out into the popular press because this is very exciting and is called a second genetic code. So you see an article from the New York Times in 1988 that is saying a second genetic code has been deciphered, solving a protein synthesis puzzle. Then fast forward nearly 20 years, and a second article in 2006, scientists say they found a code beyond genetics and DNA. So another article now talking about a second genetic code, and this was the code about the nucleosomes and the spools and so on, and they're saying the discovery, if confirmed, could open new insights into the higher order control of the genes, like the critical but still mysterious process by which each type of human cell is allowed to activate the genes it needs but cannot access the genes used by other types of cells. And that is part of the function of the nucleosomes because they allow the proper segments of DNA to be unwound to make, for example, what a liver cell needs versus a pancreatic cell versus a nerve cell versus a heart cell versus a blood cell. So every few years, we will hear about a second genetic code. And so I just wanted to share with you Edward Trifonov's take on all of this because he, of course, was there from the beginning. And so he wrote an article in 2011 in Genomics, Proteomics, and Bioinformatics saying 30 years of multiple sequence codes. So let's just read a little bit together. He is saying an overview is presented on the status of studies on multiple codes and genetic sequences. Indirectly, the existence of multiple codes is recognized in the form of several rediscoveries of second genetic code that is different each time. So each time a new discovery is made, it's dubbed the second genetic code. He's saying a due credit is given to earlier seminal work related to the codes often neglected in literature. The latest developments in the field of chromatin codes are discussed, etc., etc. And so a section in that paper by Trifonov is very amusing. He's asking how many quote unquote second genetic codes. And then he says, according to the media sympathetic to science and enthusiastic about sensational discoveries, the quote unquote second genetic code, as it was called by the New York Times, was discovered by Yaming Hu and Paul Schimmel and published in Nature in 1988. This is referring to that 1988 New York Times article that I showed you. It was about recognition of tRNAs, transfer RNAs, by respective amino acyl tRNA synthetases. We don't have to worry about the technicalities here. But 13 years later, the journal New Scientist announced the second genetic code discovered by Genuine and Alice and published in the journal Science. And again, you see all the major discoveries, Nature, Science, etc. And this time it was about histone modifications. 5 years later, the New York Times again reported about, quote, a second code in DNA in addition to the genetic code, and that was the other New York Times paper I showed you. And this was already the third second genetic code discovered by Siegel et al., suggesting the nucleosome positioning rules. And one surely would raise eyebrows having learned that there's also the fourth second genetic code on interaction specificities between proteins and DNA, and the fifth second genetic code, the name given by Nature magazine to the set of rules governing gene splicing. Bewildered reader naturally would say, I'm done with seconds, can I have a third? The conclusion from the above is obvious. One has to admit that the genetic sequences carry many different codes. If we are to know what the sequences are about, we have to detect and decipher these codes. The time of surrender to junk and selfish DNA are over, and non-coding becomes a misnomer. So again, you see how nicely this topic overlaps with both the topic of optimality of the genetic code that we've already discussed and the myth of junk DNA that we've discussed, because not only does the DNA carry these multiple layers, that same level of optimality that we talked about is present in each and every layer of information. So we have a multi-layered code that has optimized all of the layers.